Thanks, Miranda. Hello, everyone, and welcome to Novogene's Beginner Guide webinar series. My name is Naira. As Miranda introduced, I'm a senior sales manager at Novogene based in Germany, and I'll be your host for today's session. And I'm delighted to see so many of you were able to join us today, despite of the holiday season. I'm excited to guide you through the first part of our program and then hand over to my colleague Arman a little later. Arman will be sharing more about Olink proteomics and how it can be used for biomarker discovery. But before that, I would like to quickly introduce you to Novogene, who we are and what we offer. Okay, let's start on a global note. Novogene was established in 2011, and since then we have grown immensely. We currently operate 10 labs worldwide, serving around 90 countries across 6 continents, which means wherever your location is, we're here to back your research with local support and our global expertise. Zooming into Europe, we're proud to have 2 key labs, one in Cambridge, UK, right in the heart of the most vibrant science clusters in Science Park, and another one in Munich, ITB, in Germany, located in an innovation-rich biotech hub. These labs gives us the speed and flexibility to support your work closely and more efficiently. We're not just labs, though. We also have a dedicated local sales, technical support, project coordinator team, product management team, and we're right here where you are. Most of us speak your language and understand your research environment. We're here to help you tailor your research projects in the right multi-omic and offer right multi-omic solutions, so you never need to do it alone. So what does Novogene do? We are one of the leading next and third generation sequencing partners. We offer solutions for various fields of science, from research, oncology, to food and water tasting, you name it. It can also be, for example, like for forensic research and so on. All of these are grouped into service packages that can be distinguished between genomics, transcriptomics, epigenetics, and proteomics. So your one-stop shop for multiomics. And speaking of proteomics, later in this session, just a reminder, my colleague Arma will dive into Olink Reveal and explore HT panels. So 2 powerful tools that extend our multiomics offerings even further. Lastly, our multiple sequencing facilities are equipped with Illumina, PacBio, Oxford Nanopore technologies, which gives us the largest sequencing capacity in the world. Now let's take a closer look at the sequencing platforms that we have. You can see it here. We continuously invest in the very latest technologies to make sure researchers have access to the right tool for every project. Our fleet includes Illumina systems like NovaSeq X Plus, which delivers both high throughput and reliable quality, as well as long-read options such as PacBio and Oxford Nanopore. Oh, and not to forget to mention that we also do have an automation platform named Falcon. These are fully automated intelligent NGS systems built in-house. Falcon 2 and the latest Falcon 3 streamline everything from sample extraction and QC, so quality check, to library preparation and pooling. They're fast, compact, and ultra-reliable, which minimizes our sample contamination, gives us the possibility of traceable workflows, dramatic reductions in manual steps, while boosting the throughput. It's part of how we ensure consistent, high-quality data delivery. Having this range under one roof means we can support everything from large population studies requiring deep coverage to projects where read length and structural accuracy is the priority. And because our workflows are fully integrated into our Falcon automation system, we're able to combine speed, consistency, and high data quality at a scale. So innovation isn't just our tech, it is in our impact. Novogene holds over 70 NGS-related patents in more than 400 software copyrights. Every year, we process over 3 million samples. This resulted in more than 30,000 publications so far, with a total impact factor exceeding 200,000. I think that's a real scientific trust shown by our customers. So why team up with Novogene? The reasons come together pretty neatly. We deliver consistent, reliable results. Our automation and scale mean we can handle large, complex studies without breaking a sweat. We stay ahead with the latest technologies, whether it's Illumina, PacBio, or Oxford Nanopore. Our fast turnaround times outfit for speed, often faster than local core facilities, and our bioinformatics team, besides our local team, deliver data that's publication ready, helping you get to insights and papers faster. With more than 10 years anchored in Europe, we're proud to be a trusted partner for scientists like you. With this, I would like to wrap up my part of today's introduction. I hope that gives a clear sense of who we are, what we offer, and how we support your research. Now I'm delighted to hand things over to my colleague Arman, who will dive into biomarker discovery with Olink and explore HD panels. Another exciting chapter in our multi-omics story. Arman, to you.
Thank you for sharing, Naira. I'd share my screen quickly. Just a minute.
You can see the screen now.
Okay. Do you see the normal view or do you see the presenter view?
The presenter view.
Okay. Just a second. Don't know why I don't see the option for. Yeah, I think now it should work. Perfect. Cool, yeah. So sorry for this technical delay. Thank you for joining us for today's webinar. And today I'm going to talk about unlocking proteomics with the help of Olink panels, specifically reveal and explore HT. And we'd look at how these panels can help us in advancing biomarker discovery. So I will be starting with a quick introduction to proteomics. This is going to be followed by an introduction to Olink panels and the underlying principle. I will then highlight some key aspects to keep in mind while you run these panels, which include sample requirements. I will then move on to the advantages and applications of these panels, which includes biomarker discovery. And finally, I'll talk a little bit about the bioinformatics analysis that Novogene can offer to aid your research. So starting from the basics, so proteins are essential molecules that drive nearly all biological processes, be it, cellular structure to signaling to disease mechanisms. And when we talk about the proteome, so the proteome is the complete set of proteins produced in an organism, system, or biological context, which means, for example, that we can talk about the proteome of a species, for example, humans, or you can talk about the proteome of an organ, for example, the liver. Now, this proteome is not constant. It shows cellular heterogeneity, so it changes or differs from cell to cell, and it also changes over time. To some degree, the proteome reflects the underlying transcriptome. However, protein activity is also modulated by many other factors, in addition to just the expression level of the relevant gene. And these proteins show a lot of complexity because they can undergo several modifications to give rise to isoforms. And given their dynamic nature, and given the fact that a lot of proteins are present in very low concentrations or they are produced for a very short time, uh, we have the challenge of not being able to study all the proteins in great depth. However, studies in proteomics are extremely relevant because most of the targets of therapeutic drugs are actually proteins. So now let's move on to proteomics. So proteomics is the large-scale study of proteins, which includes their expression, structure, studying their functions, their interactions, and this can be within a cell, tissue, or organism. So the field basically aims to characterize and quantify the complete set of proteins or the proteome, which is present in a sample under given specific conditions. So there are two broad approaches of proteomics. One are untargeted or discovery-based approaches. So these are broad, unbiased approaches that can help you profile all detectable proteins in a sample. So these are particularly useful for identifying novel proteins. So the most popular example would be protein mass spectrometry, which is sort of the gold standard for detecting and sequencing proteins in the field. So the next set of technologies are targeted technologies or targeted proteomics. So here we have we are focusing on specific set of proteins, and we are trying to do quantitative analysis of these specific proteins of interest. These technologies, they offer us high sensitivity, reproducibility, and can also help us validate candidate proteins. So an example of this would be ELISA, which is routinely used in clinical diagnostic setups. And also Olink is actually a targeted proteomics technology. So with this, now let's talk a little bit about Olink and their panels. So Olink focuses on advancing proteomics in collaboration with the scientific community. They are based in Sweden, and they have solutions for targeted proteomics. Their technologies enable sensitive and accurate protein quantification to support research into human health, disease mechanisms, and complex biological processes. So, in terms of their products, they have assays or panels. So, these are multiplexed assays which are designed for protein biomarker measurement. And these panels, they allow simultaneous analysis of many proteins from limited sample material. These panels can be widely applied or used in different setups, be it biomedical, translational, or clinical research settings. Importantly, these panels are based on their proprietary proximity extension assay technology. So I would now introduce you PEA or proximity extension assay technology. So PEA is the principle behind the assays from Olink. So this is an antibody-based assay for protein detection and quantification. So each target protein in your sample, for example, here it's represented by this yellowish circle. So each target protein is recognized by a pair of antibodies. And each antibody actually carries a unique DNA oligonucleotide. So when these two antibodies bind in close proximity, these DNA oligos hybridize. This hybridized DNA is then extended by a polymerase reaction, and it creates a unique DNA barcode. This DNA barcode can then be amplified and quantified by qPCR in case of the small panels, or NGS in the case of Reveal and Explore HT, to help measure for protein abundance. Now moving on to the workflow. So as I mentioned before, the principle is the proximity extension assay technology, PEA. So the workflow begins with that reaction. So it begins with an immune reaction wherein the antibodies bind to the target proteins in your sample. Like I said, these bound antibodies, they have DNA tags that enable DNA hybridization when in proximity. This is followed by extension and amplification through PCR. And index sequences are added for sample identification. And later on, we prepare the libraries. So finally, after the libraries are prepared and the library quality control is done, these proteins are actually, these tags, these barcodes are sequenced using NGS. And this then provides a proxy for measuring the proteins or quantifying the proteins. So Novogene Europe is currently offering two panels from Olink. One of them is Explore HT, which covers more than 5,400 proteins, and the other is Reveal, which covers around 1,000 proteins. So I would now move on to talking about the kind of sample types that can be accepted for these particular panels. So importantly, the most common sample types for these panels would be human plasma and serum. So I want to highlight that the Olink panels that we offer, Reveal and Explore HD, they are designed for humans. And they're particularly good for plasma and serum samples. And the reason is that plasma and serum, which come from the blood, they are particularly relevant because it's one of the largest and deepest versions of the human proteome that is present. So you can identify a lot of proteins from blood samples, and this is also very relevant from a clinical point of view. Just to remind you, plasma is the fluid component of the blood, while serum is plasma without clotting factors such as fibrinogen. So when you extract blood, please either isolate the plasma or the serum before shipping the samples. So there are some best practices for sample preparation when it comes to human plasma or serum. I won't go through them in details, but one of the important things to keep in mind is to avoid hemolysis. So hemolysis is the breakdown or destruction of red blood cells that leads to release of hemoglobin and other components into the surrounding fluid, such as plasma. And this can interfere with assay results. So it's important to try and avoid it and to perform the initial steps more quickly. Also, it's important to keep in mind that all the samples are collected, processed, and stored consistently following the same protocol. Now, this assay is not just limited to human plasma or serum, but we can also process other sample types coming from humans. But the main point is that these samples need to be in a liquid, fluid, or solution form. We cannot accept solid samples, and we cannot accept samples which have a lot of, let's say, sedimentation or particulate matter in it. If you are working with tissues or you have cells or cell pellets, then these can be converted to lysates for use in the assays. If you are working with human cell lines and you want to look at cell culture supernatants, these can also be used for the assays. Now, for these other sample types, like be it, let's say, cerebrospinal fluid or cell culture supernatant or tissue lysates, for these sample types, we have to follow some recommendations and guidelines from Olink. These guidelines are there to ensure that there are no interfering substances in your sample and that your sample can run successfully. So if you're working with these other sample types, please reach out to us for suggestions on the sample type of your interest. So let's say if you're working with CSF, cerebrospinal fluid, you can reach out to us and we can send you some guidelines that you can follow while you prepare the samples. As far as human plasma or serum is concerned, we can accept these samples without any problem. We recommend that you send 50 microliters or more in 96 well skirted plates or 1.5 ml Eppendorf tubes. And the transport has to be with dry ice. It has to be a frozen delivery. So I was mentioning that it's important to prepare the samples carefully because there are a number of factors that can compromise the quality of the Olink assay. So some of these factors include use of lysis buffers that contain high concentrations of denaturants above the recommended levels. If there is excess EDTA, that can also impact assay performance. Presence of urea and urine samples can impact the assay, so it's recommended that those samples are diluted. And as I have mentioned, heme contamination, which comes from hemolyzed samples, can also interfere with assay results. Apart from that, there can be other considerations. For example, there are some samples that contain lots of proteases, So they might have to be treated with protease inhibitors before they can be used for the assays. Now I will speak a little bit about the design of the assay. I'm sorry that the slide is very busy, but I will try to walk you through it. So the initial parts of the assay, the initial steps, this PEA immune reaction, that takes place in the 96 well plates. And in 96 well plates, 10 wells are reserved for controls from Olink. So with each plate, these 10 wells are compulsorily run. So per plate, we can run 86 samples from which are coming, let's say, from your side. Now, for these 10 compulsory control samples from Olink, so these controls include sample controls, which are run as triplicates on each plate. They help to assess potential variation between runs and plates. We run plate controls that are there are 5 replicates for each plate. And this is to normalize each assay and to compensate for potential variation between runs and plates. We also run negative controls, duplicates on each plate, and they consist of buffer run as a normal sample. And these are used to monitor any background noise, which is generated when DNA tags come in close proximity without proper binding to the appropriate protein. So the negative controls are important because they assess potential contamination of assays. Apart from that, to the rest of the 86 samples, we add 3 types of internal control. So in each well, whether it's the sample well or whether it's these 10 control wells, further 3 internal controls are added, which includes the incubation control, which is basically a non-human antigen measured by PEA to monitor potential technical variations across all stages of the reaction. Then we have extension control, which is an antibody linked to a fixed pair of DNA tags that are always in close proximity. And this produces a constant signal regardless of the immune reaction. So this control helps to monitor variation specifically in the extension and amplification step. Finally, we have amplification control, which is a preformed double-stranded DNA amplicon that generates a signal without requiring proximity binding extension. This control monitors the amplification as well as the sample indexing steps. So all these controls are run compulsorily with all the plates, and these are important because they help us to do the data quality control when we analyze the data. Now, in terms of the sequencing strategy, we use Illumina sequencers, the NovaSeq X Plus in the case of Reveal currently, and NovaSeq 6000 in the case of Explore HD. Flow cell sequencing is carried out, and usually one library is sequenced per flow cell. Given these particular specifications, we can estimate close to 14 million reads per sample in the case of Reveal, and 40 million reads per sample in the case of Explore HD. But I want to highlight that this table is for reference only, and the specifications may vary depending on the project. So depending on the sample number, sample type, we might have to change the sequencing strategy. We might have to use a different sequencer, and also the number of reads per sample are also then going to vary. So this is something that you can discuss with us beforehand. So what are the advantages of using these panels from Olink? So first of all, they offer high multiplexing capacity. So you can measure thousands of proteins simultaneously from a small sample volume. For example, in the case of Explore HD, you can look at close to 5,400 proteins. They offer high throughput. Multiple samples can be processed simultaneously. So in just one plate, we run 86 samples, and we can run multiple plates together. And because we are using industrial grade sequencers from Illumina, So we can further increase the throughput also at that stage. Then the sample requirements are minimal. You need few microliters of plasma or serum. It offers high sensitivity and specificity because it is also in some sense ELISA-like. It's based on this antibody-antigen interaction. So while the specifics are proximity extension assay technology, but still because it still uses these antibodies and it uses two antibodies, it helps to increase the sensitivity and specificity. So this data is quantifiable. You can do relative quantifications. And importantly, it offers wide dynamic range. So these panels can help you detect proteins across broad concentration ranges. So you can detect as low as femtogram per milliliters. So this is particularly important because majority of the proteins are found in extremely low abundance. So out of 12,000 separate proteins that you can find in plasma, the top 10 proteins account for more than 90% of the total protein volume, while the others account for only 10%. So this is a big challenge. But thanks to technologies like Olink, you can actually detect a lot of these low abundance proteins as well. So one of the applications of Olink is in terms of detecting biomarkers. So I would first like to introduce the concept of biomarkers here. So a biomarker, a protein biomarker specifically, it's a measurable protein molecule that can act as an indicator of a particular biological state, condition, or process. This could include the presence, progression, or response to a disease. And not just that, it can also be for normal physiological events. Now, biomarkers should show some key features. So for example, they should be measurable. Biomarkers should be a measurable indicator. So they should be, we should be able to detect them and quantify them using various proteomic or immunoassay technologies, and particularly in samples like blood, saliva, or tissue, because they are particularly clinically relevant. So the second point is that biomarkers should be clinically relevant to help in diagnosis of disease, to monitor their progression, and to assess response to treatment, as well as also to predict outcomes. Biomarkers, one of the features is that they have very versatile applications. So they can be used in diagnostics, prognostics, personalized medicine, as well as clinical trial selection. Now, there are actually many different kinds of biomarkers, and they can be classified in different ways. So we can have diagnostic biomarkers, which can aid in detecting or confirming a disease. For example, PSA is a biomarker for prostate cancer, or CRP is used for inflammation. We can have prognostic biomarkers, which predict the likely progression or outcome of a disease. For example, Ki-67 could be a prognostic biomarker in the case of cancer. Then there are predictive biomarkers, which signal likely responses to a specific therapy. So now that we have talked about biomarkers, let's see how Olink can help you to identify biomarkers in the samples that you're working with. So if we talk about Olink Explore HD, it is a very comprehensive panel. It has over 5,400 validated proteins, and it can help deliver broad actionable biological insights. And the proteins that have been chosen for this panel have been carefully chosen so that they are relevant in terms of in the terms of the perspective of a biomarker. So these key features that I've highlighted here, a lot of proteins that are covered in these panels, they show these key features. Apart from that, it offers comprehensive pathway analysis. You can achieve 100% coverage of top-level biological pathways, which are critical to human health and disease. The sample requirements are minimal, which supports efficient high-throughput workflows with NGS, and it is optimized for discovery. So it's engineered for specificity, scalability, and reliability, which makes it ideal for biomarker studies of any size. And this is perfectly highlighted by the fact that Olink Explore was actually used in the UK Biobank Pharma Proteomics project, and thousands of participants, the samples from thousands of participants were processed through Olink to gain several interesting insights. Now, coming to the second panel, which is Olink Reveal. So Reveal helps in deep profiling of inflammation. It's a slightly more focused panel, but still it offers a broad proteome coverage. So you have 100% top-level pathways and 64% of all pathways in reactome. So it's still a very good panel and covers all the major pathways of interest. Like I said, it's particularly suited for inflammation. So 537 inflammation proteins, which cover 96% of immune response pathways in reactome, are covered in this panel. So the curated library of Olink Reveal has a lot of these robustly detectable proteins, which are enriched with cis-pQTLs that can help you for these proteogenomic association studies. Also, it can help in disease research. As we can see, it can help us understand or study diseases of the digestive system, respiratory system, circulatory system, and others. Finally, I want to highlight that this is a cost-effective solution. So with minimal costs, you actually get deep insights into a lot of proteins. So if I have to summarize the applications of Olink panels, so the applications are broad. They stretch across biomedical and translational research with a particular focus on human health and disease. Now, of course, we talked about biomarker discovery and validation. It can help identify and confirm protein biomarkers. So that is one of the key applications of the Olink panels. But apart from that, it can be used for disease research. You can profile proteomic changes across several diseases. It can be used in drug development and drug research. It can enable precision medicine. It can be used in clinical studies to link proteomics with genetics, environment, and other outcomes. Finally, from a systems biology perspective, you can integrate this Olink data with other data sets to get some multi-omic insights. For example, you can combine this with RNA-seq. So with this, I will then move on to the data analysis part, which is going to be the final part of this webinar. So now in terms of bioinformatics analysis, Novogene can support you with the BI analysis. So I want to give you the overview of the workflow. So from the sequencer, we get the raw NGS data, which is going to be converted into these BCL FASTQ files. And for Illumina platforms, we use the NGS2 count software to derive counts from these particular files. So this data is processed to produce these counts and layout files, which are used to build an NPX map. So NPX is actually standing for normalized protein expression. It is a unit from Olink, which is in the log2 scale. And NPX is used for quantification of proteins. So it's like a unit to quantify the proteins. In general, a higher NPX value would refer to a higher protein abundance. It's important to keep in mind that these NPX values should only be used for relative quantification, which means that they're only comparable for the same protein across samples within the same run. If you have batches, then you might require bridging samples for cross NPX run comparisons. So once we get this NPX data, it undergoes data quality control, normalization, and then we do finally downstream analysis for biological interpretation. Now, this downstream analysis is varied. So we provide you with protein expression analysis. So the quantification of the proteins in different samples. If you have different sample types or sample conditions, we offer differentially expressed proteins analysis. Then we provide functional analysis or enrichment analysis for these differentially expressed proteins to identify significantly associated biological functions or pathways. And for this, we check in the GO, KEGG, reactome enrichment, disease ontology enrichment, and interpro enrichment. Finally, we also offer protein-protein interaction analysis or protein-protein interaction network analysis. And these PPI networks are constructed using the string database. And you can also import this later on into Cytoscape for visualization and modification. So all these results are provided to you. We provide files, we provide tables, Excels. There is a report with details of all the methods. And very useful is the fact that we provide a lot of images, diagrams, and plots. So these are just a few of them from our demo report. These are just a few demo results. So we offer a lot of visualizations to help interpret protein analysis results. So this is just a selection from our demo report. And if you're interested in our bioinformatics analysis offerings, please do reach out to us and we would be happy to share more details. With this, I will come to the last slide of the webinar, which is to summarize what we covered so far. So from the service side, we offer two panels from Olink, that is Explore HT and Olink Reveal. Both of these panels are for human samples only. I want to highlight that our service is end-to-end. So you can reach out to us. We will help you with pre-study discussion. We can share relevant material, relevant literature with you that can help you with sample preparation. Then we will help you with sample shipment. We do the entire assay in our lab, which includes library preparation. We do the sequencing. And finally, we can provide you with data. And a lot of our figures are actually publication ready. In terms of sample types, this is primarily designed for human plasma and serum samples, and those samples can be accepted without any problem. The sample requirements have been mentioned here. If you're working with other samples, which would be other human body fluids, please consult our teams before you start sample preparation so that we can send you the right guidelines to follow. So with this, I will end for today, and I'd be happy to take up questions.
Hi, again. I'm sorry, I'm having some problems with my video. I hope you can see me. Thanks, Arma, for the great overview for Olink, Reveal, and Explore HT panels. We do have some questions, but please continue posting some questions in our Q&A part. If we don't get the chance to reply to them today, our dedicated contact person will get back to you with further answers. So to start with the questions, the first one that we have here is, I have 20 microliters of serum samples. This is below your sample requirements. Can my samples be accepted?
Yeah, your samples can still be accepted. We have recommended 50 microliters just to be safe in case the samples have to be rerun or you need some additional data. But otherwise, we can accept 20 microliters. The assay needs only a few microliters. So from the samples that you sent to us, we first prepare a sample plate, which requires about 10 microliters. And from that, then we prepare the plate for performing the assay. So even if you can send us 15 to 20 microliters of human plasma or serum, we would be able to accept it.
Then I move on to the next question. I want to send the samples in batches. What can be done to reduce the batch effect?
Okay. So if you're sending samples in batches, so one strategy is to have some bridging samples. So you can keep some samples common between different batches. And then based on those bridging samples, we can do normalization. So that is one. If your study is very well randomized, then intensity normalization also works. So actually, for Olink, there are different kinds of normalizations that can be done. So there's like intensity normalization, there is plate control normalization. So this combined with having these bridging samples can help correct for some of these patch effects.
Thank you. The next question is, can I detect PTMs or proteoforms with these panels?
We cannot detect proteoforms or PTMs with these panels.
That was fast. How can I obtain a list of proteins covered in this panel?
Okay, so the list of proteins can be accessed on the website of Olink. So the links are also on the slide deck. So you will get the slide deck and you can access those links to check the protein list. If you cannot find it, please reach out to us and we can send you the Excel file with the list.
Moving on to the next question. Considering the sample is run in 96 cell plates, do I need to send 96 samples? This is a good question, actually.
Yeah. Actually, yeah. So the assay is run in 96 well plates, but like I told you, 10 wells are fixed for. So I can probably just move on to that particular slide. Yeah. So 10 of these wells are going to be for controls from Olink. So in a 96 well plate, we can only run 86 samples. But if you don't have 86 samples, you don't need to worry. Our minimal sample requirements are 20. So if you have 20 or more samples, we can accept it.
Next question is, can Olink provide amino acid sequence of the protein?
Again, a good question, but Olink cannot provide the amino acid sequence. As you must have noticed in the workflow, we ultimately detect the barcodes, the DNA barcodes, and that's what we read. So we cannot read the amino acid sequence of the protein. If you're interested in the amino acid sequence, you'd have to go for techniques such as tandem mass spec.
Mhm. And we have two questions. I will join them together. It's about different type of source species. So is it compatible with mouse samples like mouse plasma and or also bovine or buffalo serum?
So these panels are specifically designed for humans, and we would not recommend to use these panels for other species because they're all mammalian species and the protein structure is similar. So it is possible that we might be able to detect some of those proteins using these panels. So if your aim is to, do a pilot experiment or an initial experiment to get an idea, maybe this is something that could be potentially considered. But otherwise, it is specifically designed for human samples, and our recommendation would be to not use it for any other species.
Then moving on, Anna is writing, I might have missed it. I would like to know the length of the reads that are required or you are providing for Olink panels.
Yeah. That's also a good question. So for this, the sequencing strategy that we're using at the moment is single end 24 because we just need to read that barcode. So you don't need to sequence the entire library. So SE24 would be the strategy for the time being.
Thank you. And I think the last one for today's session would be how many samples per condition do you recommend when we have a large cohort?
That's also a very good question, and I'd say more the merrier, of course. And these panels are actually designed to process hundreds of samples together. So you can have many patient samples. But if I have to give you a lower limit, then it is recommended that you have at least 20 per condition for more meaningful statistical analysis. So 20 or above per condition would be good.
Okay. Thanks so much, Arman. As I said, if we didn't answer your question, we will get back to you later via email. For now, I would like to thank you all once again for joining today's webinar. I hope you found it useful. A few notes. So we will send out to you the webinar slide deck with the recording sometime within the next week. You would also receive a survey today after the webinar that we greatly appreciate if you can fill out this survey, because we use this information for improving our webinars and making it more useful for you, so you get more relevant information from the webinars. Additionally, I want to mention that all the attendees today will receive a 15% discount for your upcoming project. This will be sent out to you via email as a code, and you can use it for the next project you do with us. If you have any questions or if you have any projects that we can help, please visit our website. You can find contact information there to reach out to us, and based on where you are located, you will have your dedicated account manager, salesperson contacted you with additional information. Also, if you need the certificate for attendance for your studies, this can be, you can receive this via an follow-up email, but you would just need to print out it from your browser window. Thanks again for joining us. If you can follow us also on Twitter or LinkedIn to keep up to date with our future webinars, offers, and news, and you can always subscribe for our newsletter. Thanks again, and we look forward to seeing you at the next webinar, and I wish you a nice day, afternoon, evening, or morning, depending where you're located.